Welcome to biologyexamsforyou.com. In this video, we are going to discuss about a topic, Agrobacterium, the natural genetic engineer. Agrobacterium tumefaciens is a gram-negative soil bacterium that causes crown gall disease in dicot plants. Agrobacterium has the innate natural genetic engineering capability of integrating its DNA into plant genome, ultimately resulting into a disease which is called as crown gall disease. Now let us have a look into the reason behind this disease. Agrobacterium, apart from its bacterial genome, it has a plasmid which is called as tumor-inducing plasmid or plasmid that is responsible for causing crown gall disease. Then now the organization of the plasmid, it is a 200 kb large plasmid, has four major regions: a tDNA region or transfer DNA region that has genes for auxin, cytokinin, opine, etc. that will be integrated into the plant genome. Then tDNA has a left border and right border repeats. Then there is an origin of replication. Then a virulence region that has genes that mediates tDNA transfer, and an opine catabolism region that has genes for opine utilization. So in this association, the advantage of Agrobacterium is it is actually integrating the tDNA region into the plant genome, so that the plant will synthesize opine, a nutrient that is required for Agrobacterium growth. This is the normal infection procedure. Agrobacterium tumefaciens, in the wound site, as a wound response, these bacterium get in close contact with the root site. Then the plasmid gets integrated, transferred. The tDNA portion of the plasmid gets transferred and ultimately integrated into the plant genome. And this plasmid has genes that causes proliferation, ultimately resulting in the formation of crown gall, which is all the cells are capable of synthesizing opines. That is actually a food for Agrobacterium. Now let us have a look in detail into the plasmid organization. First of all, the most important region, which is the transfer DNA region or tDNA region. This region consists of three genes: auxin, a cytokinin, a region that encodes for cytokinin, and an opine encoding region. Auxin and cytokinin gene induces cell division. That is as a result, crown gall appears on the plant. Then opine gene will synthesize opines. That is not at all needed for the plant. It is just the nutrient that is required for the bacterium. Then there is a left border and right border repeats that are required for successful transfer. So this is the region. The region between the left border and right border repeats, that is the transfer DNA region, is integrated into the plant genome. Now another one of the most important second part of the plasmid, which is called as the virulence region or vir region. In this region, a number of genes are present, and these genes are organized into 8 operons designated as vir A to vir H, and has approximately 25 genes. And vir region mediates the transfer of tDNA to the plant genome, and this genome actually regulates the transfer of tDNA and successful integration into the plant genome. Now let us look into the detail, the process of infection, the stepwise process. A wound occurs in the plant. A wounded plant region produces a chemical which is called as acetosyringone as a wound response. Actually, the bacterial chromosomal genes called CHV genes or chromosomal virulence genes facilitates binding of bacteria to the wound site. Now the vir region, acetosyringone, activates the vir region binds to Vir a protein which is an acetylcholine receptor. Vir A phosphorylates Vir G, another Vir operon, which dimerizes and activates expression of all Vir operons. All the Vir genes together mediates successful tDNA transfer and its integration into the plant genome. Now this is the diagrammatic picture. You can see the plasmid right there. Bacterial chromosome has CHV genes that facilitates binding of bacterium to the plant cell, intimate contact of bacterium to the plant cell. Then Vir genes that is mediating the transfer of tDNA, T-strand. There will be a conjugation tube formation. Through that tube, the tDNA strands is passed on to the plant cell where it moves to the nuclear pore complex and later gets integrated into the plant genome. And what is the advantage or how we exploit Agrobacterium's natural genetic engineering ability in our gene cloning technology? And as we mentioned in the second slide, this is the tDNA. We are introducing the gene of interest into the portion of tDNA, or any fragment can be transferred into the plant genome by replacing tDNA with our gene of interest, provided that the essential prerequisites are left border and right border repeats should be there. So any gene that is placed between left border and right border repeats will be transferred and integrated into the genome. Then Vir regions are required that is responsible for transfer. Then the third thing is the chromosomal genes or CHV genes, virulence genes are required that will facilitate the binding of bacterium, intimate contact of bacterium to the plant cell. And these are the three prerequisites for a successful transfer. Now this is a diagrammatic sketch. We have a genetically engineered plasmid which is called as disarmed tumor inducing plasmid, where the tumor causing genes like auxin, cytokinin, etc. are replaced by with our gene of interest. So this plasmid is this genetically engineered Agrobacterium is co-cultured with tobacco plant cell, maybe protoplast. Later transformed cells are formed. From these we are regenerating a plantlet. Now we have a transgenic tobacco plant with our gene of interest in all the cells. For more detailed notes, visit www.biologyexamsforyou.com. Thanks for watching.